Okay, hi everyone. I'm going to try to get going if that's okay. We have a lot to cover today, and I want to also make sure that I cover what you need to know for your tutorials on Monday, and I also wanted to make sure that you had a good idea of how I'm going to be testing you on the midterm, and I'll leave that right to the very end. Today's lecture is actually fairly dense, and there's a lot that you're going to be tested on. I don't know if any of you actually found the little hidden Easter egg about this particular lecture. It is there. I'm going to be highlighting a couple of other things on your announcements page and on some of the other pages that we have. Hopefully you'll be able to find those and share them with your colleagues if you do find them online. I'm going to go back and tell you a little bit about one of the more interesting stories that is developing right now in terms of what we think might be related to diet. And today is actually the last lecture that we're going to have related specifically to obesity. We're going to be switching gears and looking at some other factors starting next week on the microbiome, how your diet is impacting the microbiome and how it might lead to obesity. But today is the last sort of molecular phenotype on obesity. And then on Monday, you'll also have a tutorial based on why we eat this types of things that we are eating right now. Today I want you to focus a little bit differently. When you're taking down your notes, when you're jotting things down that you're going to come back and study over the weekend, I need you to focus a little bit differently than you have been in the past. I need you to understand some of the details that are related to the neurotrophin system as well as their receptors. I'm going to give you a little bit of detail about the P75, and you should jot down a couple of things about P75. And then I'm going to jump into the cyclic AMP protein kinase A pathway. We had actually talked about this pathway in the last class. And in the last class, in the adipocytes, when you activate the cyclic AMP protein kinase A pathway, that causes lipolysis. And we talked about that in the context of leptin and how leptin signaling then causes lipolysis through this pathway. And I'm going to go back over this in a little bit more detail. So this is the paper that I'm interested in sharing with you. And this goes back to obesity. And right now, the reason why I'm concentrating on obesity, there are a number of reasons. It impacts you directly, sort of as human biology students. Secondly, it is an epidemic. And thirdly, we know that as a consequence of all of these different things that are going on, when you are gaining weight, and you will gain weight as you get older. As you're gaining weight, what are the long-term consequences? So we're not talking just about metabolic syndrome. We are talking about things like potentially Alzheimer's disease, increased risks of cancer, all of these things that you guys are going to be taking care of me when I'm a little bit older. You're going to be paying for my healthcare costs, and all of this has to be included in the healthcare policies and things that we need to take into consideration. So this really odd story about the P75 neurotrophin receptor was really fascinating because, as I mentioned in the last lecture, we have a lot of drugs that help to control the way in which we regulate food, our sense of appetite, but a lot of those drugs have some serious consequences and some serious side effects. So people are always looking for new drugs to help regulate the burning of fat, but not affecting other systems. And I'm going to try to take you through some of the rationale why this study is actually potentially really important. So this one slide, you might think, well, you know, I'm going to go back and I'm going to ignore it because he's not going to concentrate on this. This is actually an important slide, more important than you might give it credit for. So we know that there are neurotrophic factors that I'm going to describe to you in the next few slides and give you a brief review. These primarily work in the brain. So in the brain, if we have a reduction, either through transgenic means like BDNF, a heterozygous mouse, or a homozygous mouse, which doesn't survive, in case you're wondering why it's heterozygous. If we deplete the brain of BDNF, then there are changes in the way in which mice start to eat. And here it's really related to eating. These mice, if they are lacking BDNF in the brain, start to become obese. And these are central effects. These are effects that take place within the CNS, within the brain, and we know it controls how much they want to eat. And it's not related to leptin directly, it's a separate mechanism. And they gain weight tremendously. But people were looking at this receptor system inside the brain, and they realized that when they started mapping out where are all these different neurotrophin receptors, and these are some members of the neurotrophin receptors, they realized that it's not just in the brain. BDNF is not only found in the brain, it's found peripherally. So is NT3, so is NTF, so is NT4, 5. All of these things are found outside of the brain. And they started to wonder, why are all these brain, what they thought were brain specific, why are all of these receptors and the different neurotrophins, these chemical compounds here at the top, why are they found in peripheral tissues? Why do we find them in skeletal muscle? Why do we find them in other areas? So again, I need you to know this diagram for a variety of reasons. These are the different neurotrophic factors, which I am delineating as NT. This normally work on your brain. NGF is one of the characteristic neurotrophic factors. BDNF, that we'll be hearing a lot about in this course, is another factor. So is NT4-5. All of these are neurotrophic factors, and so is NT3. Generally speaking, these work on specific receptors. NGF, for example, binds to, in the brain and in peripheral tissues, to the TrkA receptor. BDNF binds to the TrkB receptor. NT4-5 binds to the TrkB receptor, but so does NT3. And some are more specific. NT3 is specific for TrkC, and some are less specific, as according to this diagram. Now, we're going to hear a lot about BDNF. Some of you mentioned in the last class that you know about BDNF. How do you deplete your brain of BDNF? Any ideas? How do I make your BDNF levels go down? What makes your BDNF levels go up, in other words? Exercise, and that's one of the things that we're going to try to look at. You know, is exercise really that important? Is exercise in the literature? Can we evaluate it, or whatever we read, that's this article that I'm showing to you, how do we know that this is right? How do we know that this is true? And how do we know if this article is a good one? And that's one of the things that I want you also to take away from this course. So again, there's a lot of detail here. Some of you really love multiple choice test questions that are detail oriented, and I'm going to tell you that yes, you'll get one based on this particular slide, that you'll get a question based on, tell me a little bit about neurotrophins. All of them, BDNF, NGF, NT3, NT4-5, all of them start off as bigger molecules, and then they get cut in half, roughly to half their size. And these long precursors get cut down to about 100 amino acids or 120 amino acids. And again, NGF was the prototypical growth factor. BDNF, that we'll be talking a lot about, is another factor that was purified, that has homology with. It's similar to NGF, but it's a little different. Only about 50% of it sort of matches up with NGF. And then neurotrophins NT3, NT4-5, through PCR-based cloning, was found to also be other members of this same family. In other words, if you have these factors within the brain or peripheral tissues, they make them grow, or they make them start dividing. And those are very useful things to keep in mind. Now, there's one other thing that I need to mention to you. All of those neurotrophic factors, BDNF, NT3, NGF, all of them, all of the ones that I showed you on the previous slide, every last one of them has the capacity to bind to this receptor. This receptor that I'm showing you here, the P75, is the topic of today's cell paper that I'm going to go through. And this P75 receptor is really different looking. It doesn't look like any of the other receptor subunits that I showed you earlier. It doesn't look like TrkA, does not look like TrkB, nor does it look like TrkC. All of these bind to the P75 at very low affinity, and that's sort of the but that you see here. We now know that this receptor is found peripherally. We know it's found on adipocytes, for example. Why is it here on adipocytes when it should be in the brain, when it should be acting on neurons? We're going to go through some of the rationale of that paper in some more detail. So again, I want you to be very clear on this. If you are lacking BDNF, you are going to gain weight because you are eating more, okay? It drives a hunger response in you. Low BDNF, you eat more and more, and that's why you get obese. Here, when we're looking at P75, it's not about eating more, it's about burning fat. If you have or don't have P75, you are going to change how your fat cells are responding. Okay, so all of these neurotrophins, the BDNF, NGF, NT345, and their receptors are also expressed in a number of metabolically active tissue, including adipocytes. And therefore, they might be important in regulating weight gain. So in the brain, BDNF controls whether you're hungry or not. And we have all of these other neurotrophins outside of the brain. The authors of this current paper wanted to see what is their role here in obesity. So one of the things that they found right from the get-go is that if you don't have P75, that low affinity neurotrophic receptor, if you don't have it, you actually don't become obese. And that was weird. It was a very strange, unexpected result that they had. They had no explanation for it. It was just an observation originally. Not only do they not become obese, even if you give them a high-fat diet, which is going to be abbreviated as HFD, even if you give them a high-fat diet, they also don't become metabolically or have metabolic syndrome. And so we know that this is playing a unique role. It's nothing about food intake that I'll show you in a moment. And one of the things that they found was that if you do not have P75 neurotrophic, the neurotrophin receptor, this is actually going to control how quickly you burn fat. If you don't have it, you burn fat quicker. Okay, so this was an unexpected finding. If you knock out BDNF, you no longer have a brake on appetite, and they thought, well, okay, let's see what happens if we knock out this receptor. Unexpectedly, surprisingly, they found that they actually didn't gain weight. It was the opposite trend to what they were expecting. So manipulating this system, helping you burn fat, if this system is correct, that if we find the right way to trigger that response only in adipocytes, you could eat whatever you want. They ate high-fat foods, and they did not gain weight. They burned fat as quickly as they were eating. And so if we can manipulate the system, for drug companies, it would be a tremendous boon. So again, P75 is expressed in a number of tissues. They went back and checked. It's expressed within the liver, metabolically active. It's where you store glucose in the form of glycogen. White adipose tissue, again, in the adipocytes, if you have excess glucose, it gets converted into triglycerides and free fatty acids within the adipose tissue, as well as in skeletal muscle. So all the places where you'd expect to see like insulin and other things, we would also see here with P75. But they didn't know how this was working originally. I just gave you a summary snapshot diagram, and now I'm going to actually take you through some of the data. You do not, by the way, have to memorize the data, but I want you as young biologists who are learning how to interpret data and how to read papers and how to potentially present papers to have an idea of the different types of experiments that researchers do. So one of the first things that they decided to do was they wanted to see, using a Western blot, where is this receptor being found? That previous slide that I just showed you highlights the major results. So on a normal diet, which is the ND here, versus a high-fat diet after 3 weeks or a high-fat diet after 8 weeks, if they took a look at the P75 expression, they saw a big increase in the protein production of the P75 neurotrophin receptor in the high-fat diet only. And again, it was only in white adipose tissue. So they saw this big increase, shown here numerically by this bar, that after a high-fat diet in adipocytes, that they were seeing a big increase in the P75 receptor. That's the normal condition. That's what happens. When you're gaining weight, this P75 is being induced within your adipocytes. Is that okay for everyone? And these bars, again, you don't have to memorize the percentage increase or anything else. They also showed that under normal circumstances, when you look at other fat tissue and other adipocytes, that anytime you form an adipocyte, they all contain the P75 receptor, okay? So P75 is found in adipocytes. P75 is induced. It becomes much, much higher if you start eating high fat. Any questions about this before I go on? Yes, 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 question. That's an excellent question. So why do we see the increase after 3 weeks and then it goes back down to the same levels after 8 weeks? They don't really have a good explanation for that in their paper. And it's a great observation. It's one that you should point out if you're doing a journal club. But one of the things that they're thinking is that it's an induction phase that starts a metabolic change within the adipocytes, and it starts the process but isn't necessary to complete the process. So it's a great question. So if you took out P75, what would happen to these mice? And this was the question that they asked next. So in adipocytes, we have P75. If you have P75 after high-fat diet, you will increase the induction of P75 levels in adipocytes, not in skeletal muscle, not in the liver, only in the adipocytes and only there. What happens if you take out, through transgenic mechanisms, the P75 receptor, the NTR, the neurotrophin receptor? So again, they're placed on a high-fat diet. We know that P75 is going up under normal circumstances, and in wild type, we would expect it to go up. And we know that they become obese and they start gaining weight. But if you don't have the P75 receptor and you knock out the P75 receptor, even on a high-fat diet, these mice don't gain weight. They grow, which is what you're seeing there, but they don't grow above what you normally expect. On a high-fat diet, these mice on the top bar in black are actually becoming obese. What you're seeing on the bottom is that over time, the mouse is simply getting bigger. They're just developing as they normally would on a normal growth curve. So they're not becoming obese. Just by knocking this P75 receptor out on that same diet, they are not becoming obese. Now, this actually is a really important diagram for a number of reasons, because I wanted to emphasize a couple of things. The P75 knockout at the same time, over the same length of time versus a wild type on a high-fat diet, you can see the change in body size, okay? It looks a lot like the leptin deficient mice that we saw earlier. If we look at this mouse here versus the P75 knockout mouse, just out of curiosity, because you'll need to know this later for your midterm, what type of figure am I showing you in part C? How am I looking at fat in that particular figure? Yes. It could be a DEXA scan, but what does this look more like? Yes. This is an MRI. Do you remember why we use MRIs? And it is indeed an MRI of a mouse. So this MRI tells us, where is the fat going? Is the fat just going everywhere? Or as the mice gain weight through a high-fat diet, are they gaining it around the abdominal cavity? And you can see that they get a lot of fat inside, around the abdominal cavity, just like you would expect would happen in you if you had a high-fat diet. So again, we won't go through all of the individual slides here. There are a couple of things that I wanted to point out to you on this particular slide. The adipocytes after a high-fat diet. So they're eating lots and lots of fat in their diet. Of course, the adipocytes themselves are getting bigger. They have the P75 receptor. In the knockout animal, without the P75, the adipocytes don't actually get any bigger. They stay about the same size as you would expect, even with high fat. And again, even after 3 weeks on this high-fat diet, the adipocytes in the wild-type mice tend to be much, much more enlarged. You can see how much bigger they are. This is the lumen where you are storing fat in an adipocyte. Here's one adipocyte. Here's another adipocyte. Here is a third adipocyte. And you can see for the same length of time that they've both been on a high-fat diet, the wild-type mouse has a tremendously bigger enlarged adipocyte versus the one that has the knockout of the P75 receptor. And overall, this is a trend that they saw, and they wanted to know, what is the mechanism behind this? Yes, the adipocytes are smaller. Yes, the mice don't gain weight if we knock out this receptor, but what is it actually doing, right? Do you have any questions on this before I go on? And again, you don't need to really worry too much about the overall size effect, memorizing the size effect. I'd rather you concentrate on the data that's down here on the bottom. The last thing I'm going to mention to you because some of you will go on to some third-year courses. One of the ways that we can stain for these adipocytes is using a histological stain known as hematoxylin. And hematoxylin outlines these adipocytes really well. It's something that you might want to make sure that you go back and study over the weekend. If you're asked, how do you stain for adipocytes histologically? You put them under hematoxylin, and it stains the cell membrane, as you see here. So it's one thing to say that these mice don't gain weight, right? That's one possibility. They don't gain weight, but they're not obese, but they still have metabolic syndrome. And one of the things that these authors wanted to try to do is to go back and see. These mice don't gain weight, they don't look fat. On an MRI, they look normal. The adipocytes histologically look normal. But maybe they have problems with their insulin as well as glucose tolerance. And again, don't worry about the overall size effects here or memorizing the size effects. But one of the things that they showed was that these mice, after a high-fat diet, they were still responsive to insulin. If you give them insulin, glucose levels go down. So you give mice a shot of insulin and you give them some glucose, the glucose levels in the blood drop, which means that they're still sensitive to having insulin in their bloodstream. We know in metabolic syndrome that disappears. And compared to the ones that are on a high-fat diet where the insulin sensitivity is not there anymore, it's certainly still much more pronounced. We know that they also can get rid of glucose a lot faster as well. So if you give them a shot of glucose and you see how much glucose is in their blood, their tissues can respond to glucose much better. As you see down here, the glucose in the bloodstream stays relatively high. So we know that through both the insulin tolerance test, or the ITT, they are still responsive to insulin. And we know that they are also able to clear glucose from their blood. In other words, it's not that they're just not getting fat. It's not that their adipocytes are staying the same size. On a high-fat diet, mice are becoming metabolically inactive, or they have metabolic syndrome, but these knockout mice, the P75 knockout mice, actually don't. They stay responsive to insulin and they can respond well to glucose. The other thing that is very common and we haven't talked about, and you should make sure that you know for your upcoming midterm, is that when you start gaining fat or you have too much glucose in your blood that gets converted to fat, one of the tissues that gets converted really quickly into fatty tissue is not just the adipocytes, but also your liver. And your liver undergoes what is known as hepatic or liver steatosis. And this hepatic steatosis is very evident here. This is the liver that you see. This is the actual liver that you can isolate. And you can see that in the P75, it is much, much smaller. These are normal looking hepatocytes here, but the hepatocytes have been blown out. There's lots and lots of fat, which we can't stain for using this particular stain. The fat are the empty spaces that are there. This is what the normal tissue should look like on the right. So 16 weeks after, with a high-fat diet, the liver has become completely fatty. It becomes basically useless. The liver stops functioning like it normally does because the hepatocytes have been taken over by so much of the fat. And this whole process together is known as liver or hepatic steatosis. The other thing to go back and check for is that when the liver starts to become fatty, the other thing that tends to change is this regulatory protein known as SREBP, or sterile regulatory element binding protein. And when the liver starts becoming fatty, this protein starts to drop. But in the knockout animals, as the liver stayed nice and healthy, the levels remained high. And that's a diagnostic marker, which you might be asked about on your midterm, that we can go in and check. Your liver function, if it's normal, we should have high levels of this sterile regulatory binding protein. But again, how does this actually work? We don't know this yet. It's not something that I've shown you the data for. And the researchers are quite curious. None of these changes occurred. You give them high fat, they seem to be perfectly normal, no metabolic syndrome, no weight gain, adipocytes look completely all right. But why is it? Is it simply because not having the P75 works Because they don't eat. Remember BDNF, you lose BDNF, you start eating more. Maybe if you knock out the P75, you are actually changing the way in which the animals eat. However, when they started looking at food consumption in the P75 knockouts, and this is a slide that you should know the general data from. You should know that they're not eating more, or they're not eating less. They're eating exactly the same. So when they look at the mice either during the day or in the evening, one of the things that they find is that the food consumption, shown at the very top here, the food consumption, whether you look at it in graphical form or individual mice, actually, they're the same. If you normalize it for the weight gain that they're experiencing, the amount of food that they're taking in is actually identical between the knockout versus the wild type mice. So they're not eating more, they're not eating less. That's not the reason why they're not gaining any weight. However, the other possibility is you can gain weight or lose weight by changing your diet and how much you're taking in. The other possibility is on a high-fat diet, you're just simply burning off more of the fat that you are taking in. And they wanted to explore this possibility down here on the bottom. And one of the things that they showed was that indeed, they're not eating any more or less than the wild-type mice. They're eating identical amounts, which is shown here and then over here as well. But what is happening is that overall, they are much more metabolically active. Simply knocking out the P75 makes them a lot more metabolically active, both during the daytime or during light cycles, as well as in the evening when they're even more active than their counterparts. So this might be one of the reasons why all of this occurs. And when they went back and checked, this is exactly what happens. If you remove P75 from the adipocytes, all tissue, but including the adipocytes, those adipocytes are much more effective at burning off fat. So you take in high fat through your diet, you're eating just as much as someone else on a high fat diet, but now you're burning it much more effectively if you don't have this P75. And the fat oxidation, as well as the production of heat, the thermogenesis that I mentioned to you in the last lecture, in the adipocytes was dramatically increased. So they are burning off much more than they are taking in, which is what the bar graphs are showing you here on the left. Again, if you take a look, a couple of other things that are really important, some of which hopefully you'll remember from the last lecture. Not only are they burning more fat, how are they burning more fat? By lacking this the P75 neurotrophin receptor, what they're doing is they're increasing the expression of the uncoupling protein, or UCP1. Do you guys remember UCP1 from the last lecture? If you don't, that's okay, because it's been a long week. However, for your midterm, where you're going to be labeling a diagram, that's a diagram that you probably want to go back and make sure that you review. The uncoupling protein works through the mitochondria to burn fat. And what they're seeing here is that without P75, they actually have much more of the uncoupling protein that you see here. Uncoupling protein levels are much higher in the P75 knockout, and so is another sort of oxidation related protein, deionidase 2, and its expression is also much higher. When they're found in the mitochondria, it helps the mitochondria to burn fat much more effectively. Somehow, losing P75 causes an induction of both uncoupling as well as DO2. The other thing that they mentioned and they found, and this is a factor that we did discuss being released from adipocytes, is adiponectin. I mentioned to you adiponectin is really good for heart health. It's really good for preventing forms of cell death. But another thing that adiponectin does that wasn't on that previous slide from Monday is that it increases fat oxidation. The more adiponectin you have, the better your fat is burned off through beta oxidation. And what they showed was that in the P75 knockout animals, they actually also overexpress the adiponectin versus the wild type control. So everything makes it look like this animal, by simply removing P75, burns fat, doesn't accumulate fat, and it does so in a metabolic way through increasing adiponectin, increasing uncoupling protein, as well as increasing the DO2 gene expression. Do you have any questions on this before I go on? So I want you to highlight this slide for the time being. This is a slide that we're going to come back to at the very end of today's lecture. It's a slide I want you to be thinking a little bit about as well. Because we're in biology, one of the things that you can't really do anymore, you used to be able to do this in the 1970s. In the 1970s and 80s, all you had to do was show a phenomenon. You knock out P75, or P75 isn't there, the mice get bigger, or they stay the same size on a high-fat diet. That was it. Nowadays, in biology, we're required to demonstrate some kind of mechanism associated with this. So how does knocking something out actually increase the uncoupling protein? How does it increase lipid oxidation? How does it prevent you from gaining weight even when you're eating the same amount of high-fat food as someone else? Now, one of the things that you can do is use a chemical compound known as isoproterenol. And isoproterenol is a pharmacological compound that you can inject. And one of the things that it does is it raises inside the cell the level of cyclic AMP. It is a cyclic AMP agonist. It increases intracellular cyclic AMP levels. And when you start doing this, you can increase the level of lipid oxidation, or the burning of fat, and you can see the free fatty acid release as you burn fat. It can increase this by about 1.6-fold in the wild type, but it increases it even more so in the P75 knockout animals, which means that something in the cyclic AMP pathway is being enhanced in the P75 knockout animal. We get a much bigger, more robust burning of fat as released as through the release of free fatty acids if P75 is not there. So somehow when you break down lipids more effectively without P75, it involves the cyclic AMP pathway. And if you go back to that diagram that I showed you earlier on this week, you'll know that pKa, cyclic AMP, is involved in that lipid lipolysis pathway within adipocytes. So somehow this is cyclic AMP dependent. The other compound that you can actually use is this H89, and this H89 is found all the way over here. You can block cyclic AMP using this. So isoproterenol increases cyclic AMP. H89 is a drug that you can give to block cyclic AMP, block lipolysis, okay? And I can tell you, on your midterm, you're going to have to know when you use one drug, like isoproterenol, to increase cyclic AMP, and H89 to decrease the levels of cyclic AMP that you see here on this part of the diagram with this H89 application. It drops lipolysis all the way down. So we think that this is all sort of related. So somehow within these adipocytes, something is involved in the cyclic AMP pathway. Now this is a really busy slide and I have to apologize for this, but it's a really important slide for you to go back and review a little bit as well. So ultimately, cyclic AMP works on pKa, and that was on that slide on Monday. pKa itself, the thing that cyclic AMP is acting on, is a tetramer. It has 2 catalytic domains and it has 2 regulatory domains. And this becomes important in terms of why the results were shown in the way that they were. In the knockout animals, there's no difference. The levels of cyclic protein kinase A are identical. There's no change. There's no upregulation if you were thinking somehow the levels of the catalytic or the regulatory were increased after you knock out P75. However, if you knock out the P75, that catalytic subunit tends to become more active. It's no longer being bound by the regulatory domain, and so it becomes much more constitutively active. It's turned on all the time because the P75 is not there. And that's because this P75 is also shown to directly bind to the regulatory and catalytic domains. So if it's not there, and is normally acting as a brake for the interaction between the catalytic and the regulatory domains. Now, without that regulatory domain stopping it, acting as a brake, the P75 is no longer acting on the regulatory. The catalytic is free to do whatever it wants. It can burn fat as much as it wants because that's what pKa will go off and stimulate. So We know also in the literature that if you delete the regulatory domain, it looks identical to this particular phenotype that we've just examined with the P75 knockout. Do you have any questions about this before we go on? There's a lot in this particular slide. Yes, no, maybe? Any questions? Have you guys talked about PKA in any of your other courses? Yes? Yes, so you're all experts on PKA, kind of. Okay, I know it's a busy slide, and I'll try to make sure that you are as prepared as possible for this. Okay, I'm going to tell you a little bit about your tutorials on Monday. Some of you have been asking, and the rooms have been posted up for you. On the time that you signed up, whether it's 10 a.m., 11, or at 4, you will go to any one of those 4 rooms. I would suggest that if you know someone else going to the same time, go along with them. You're going to be doing a lot of group work together, so you might as well do some group work with your best friends, I guess. Hopefully it won't lead to best friends not being best friends at the end of that group work. But I want you to enjoy going and doing things together with the rest of your colleagues. As I've posted for you, you've already seen the brief article. Based on that brief article that your TA is going to make sure that you have understood, they're going to give you a brief overview, make sure that you've understood it, you're going to write a quiz. The goal is for you to get 5 out of 5 on that quiz. If you're hoping to get 3 out of 5 or 2 out of 5, we're not going to let that happen. So we want you to all do really well, and we picked articles that are sort of related to wrapping up this particular theme in this course, and it will be the same for the next theme that we cover as well. We also want to give you some ideas as to how you're going to be tested and other things. So we want to make sure that you are well prepared for this. And I'm hoping I actually have this slide right. Okay. Do you have any questions about your tutorial at all? Or anything related to what we covered here today before I just wrap things up? No? No questions at all? Perfectly clear. You're going to get like 100% on that section of the midterm. Yes? Okay. So this is how I'm going to make your midterm, and this is what I've been doing over the last couple of weeks. Is kind of getting ready for your midterm. I want to make it fair, but I also don't want to make it like completely fluffy as well. So this is a slide that we just went over. I asked you to tag this slide. What kind of questions do you think I would ask you on this slide? I'm not going to be asking you about the size effect. I'm not going to be asking you how many fold increase would you expect to see, et cetera, et cetera. But what do you think I might ask you based on this slide? Any ideas? No ideas. You must have some ideas. You know that you're going to be asked that on your midterm, right? You weren't sure? You're going to be asked that on your midterm. So what might I ask? Yes. About the function of adiponectin? Possibly. Okay, that's a possibility. I might ask you about the function of adiponectin. Anyone else want to make a guess? Yes. The P75 has a lot more uncoupling protein. Okay, great. This is what I'm likely going to ask you. So I like having sort of a little scenario. So she just finished her undergraduate studies and now is in graduate school. As part of her thesis, she has to be able to create a transgenic mouse that will increase thermogenesis. So hopefully that will ring a bell for you on the midterm. We just covered that. They burn more fat. And how might she do this if she overexpresses a gene that is related to burning through fat? So which one of these, in other words, might not be so useful? Wait, can I get in the picture? No, go ahead. It's okay. Yes. Okay. Thank you. Any ideas? I'm hoping, because this is the type of question that you're going to be asked. It's pretty straightforward. We covered it today. I'm hoping all of you have a fairly good short-term memory and an even better long-term memory. Any ideas? A, B, C, D, E. So again, which ones of these are not going to be, based on today's lecture, related to helping you burn through fat? Lipolysis, thermogenesis, oxidation. C. All right, we have gone to Spain. I like that. So you are C. You are correct. So this one here, as I mentioned, where did we find this, by the way? We find this in the liver. It's normally there in liver production. It's not. It's making sort of liver fatty. It has nothing to do with the burning of fat or lipolysis or thermogenesis. Are you going to expect to see this on your midterm? No? Actually, you'll have to wait and see. Maybe you will, maybe you won't. So next week, we're going to actually change gears a little bit. We're going to talk about not necessarily obesity in the sense of the mechanisms that might play a role in obesity. We're going to look at obesity from a slightly different standpoint. I'm actually right now, for if I can get a small crew up here, I'm going to try to save your life, or save the lives of lives inside of you. So if there is a small group of students who don't mind being embarrassed a little bit. Okay, all I need you guys to do is to line up in the order that you think which one is more expensive and which one from least expensive to most expensive. Sorry? I didn't go to Metro though. Sorry. And actually, if they take some of this away, I am actually saving their lives, because all of them have a common theme. It may not look like they have a common theme, but they all do. This is what your tutorial is going to look like on Monday. No one agreeing on things.
Is it amount based?
Just price.
Just price?
Yeah. Okay. Are you guys in agreement at this point? Okay. Sorry, it's not the Price is Right, but it's close enough. Actually, I can't remember, to be honest. The thing that I wanted to point out, and please help yourselves at the end. The thing that I wanted to point out is in next week's lecture, your diet, whether you think so or not, is actually changing your gut microbiome. And I'm not talking about the sugar intake. We know that that certainly is going to change your gut microbiome, therefore affecting your health directly. But every time you eat something that does not contain fiber, which all of these things do, even the granola bars at the end, fiber one of the reasons why they put fiber in there is because now we know that fiber is essential for your gut microbiome, as we'll cover in next week's lecture. Your microbiome impacts your health, your human biology-ness, just as much as all the things that we talked about here. And that's sort of the next module that we'll be getting into. And if you are interested in any of these fiber-containing things, I'm happy to save the lives of all of the millions to billions of bacteria living in your gut. So please help yourselves. I'll see you on Monday, and I'll see you at some of your tutorials on Monday as well. Nice.